everyone, welcome to Ibdp Biology. Today we're going to talk about cell division. So this lesson is a very important lesson and I want you to be a bit more attentive because there are a lot of details. So first, try to understand why do cells divide. For previous lessons, if you remember, we talked about surface area and volume ratio, and we learned that as the cell gets bigger, the surface area and volume ratio gets less efficient. So if an organism grows large, it needs to produce more cells. Cell division, although the growth of the organism by producing more cells and more cell differentiation. Cell division is actually called mitosis. So let's have a glimpse at the cell division, how it occurs. This is actually how the cell division looks. Those long strands are actually the chromosomes. Chromosomes are separated and the cell division occurs after the cytoplasm is divided. So this is how the cell division acts. So mitosis is actually a type of a cell division happening in eukaryotic cells. So when does mitosis take place? In tissue growth. In multicellular organisms, mitosis increases the number of cells, so the size of the organism. Asexual reproduction, for example, some organisms such as yeast, they reproduce asexually while budding, and the formation of the bud is a result of mitosis. So tissue repair, damaged tissue repair happens through mitosis. Even in human, you see that there are damaged tissues, and by the cell division this damaged tissue is actually repaired. And lastly, embryonic development. During sexual reproduction, a zygote is formed, but then zygote actually undergoes mitosis to produce the first embryonic cells and consequently the whole embryo. The cell cycle actually looks like this. First comes the interphase, where the cell actually undergoes a lot of synthesis processes and everything doubles in the cell, the phase in which the cell spends most of its time. And then comes the mitosis, a division process of the diploid eukaryotic nucleus, and then cytokinesis, division of the cytoplasm to form two daughter cells. The cell cycle is a series of events between one cell division and the next, and the result of the cell cycle is the formation of two genetically identical cells. So first we're going to look at the interphase. Interphase is actually a part of a cycle that do not involve cell division. The average cell spends 90% of its life in this phase. During interphase, everything doubles in the cell because cell has to be divided into two daughter cells, and that's why everything has to be shared equally. It has three subdivisions like G1, S, and G2. So growth 1, synthesis, and growth 2. In the growth 1, G1, the volume of cytoplasm is increased, organelles are produced, and proteins are synthesized. And in S phase, DNA is replicated. In a G2, again the volume of cytoplasm, organelles, and proteins are again doubled. So interphase process, as we said, it's actually doubling all the properties of the cell. So metabolic reaction actually going on as a normal cell. Protein synthesis, organelle numbers are increasing, and DNA is replicated to ensure a second copy is available to enable mitosis. So today also we're going to talk about supercoiling of chromosome, but first let's understand what is a chromosome. So guys, chromosome actually consists of proteins and DNA. So the proteins or DNA are so much packed to each other that they create the chromosome. So DNA with proteins of histone, then the chromatid fibers, they are so much coiled and they create the chromosome. So let's watch how supercoiling of chromosomes usually happens.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells.
So, why supercoil chromosome? As you know, chromosomes are not all the time in the cells. They are actually formed when the cell is actually starting to divide during interphase processes. So the chromosome need to be stored compactly to fit within the nuclei of the cell, because nuclei is going to divide and share the DNA of the organism equally between two daughter cells. This problem becomes more acute during mitosis, when chromosomes need to be short and compact enough that they can be separated and moved to each of the cell. So on the left top part, you see the chromatin fiber. It's made of the DNA coiled around the cluster of protein called histones. Then during S shape of the interphase, DNA undergoes the replication, and at the end of replication, each chromatin fiber has replicated. The two identical molecules are now called sister chromatids, and they are attached to the centromere. This is how it happens. So now I'm going to show you the supercoiling of chromosomes.
These chromosomes, shown at more than 6,000 times their original size, are from a dividing eukaryotic cell. During cell division, each chromosome has already been duplicated, so it consists of 2 DNA molecules that run the length of the chromosome and are tightly coiled around proteins. The coiling of DNA within cells occurs in the following way. First, the double strand of DNA winds around separate clusters of 8 histone proteins. Each DNA histone complex is called a nucleosome. As shown in an electron micrograph, the nucleosomes look similar to pearls on a string. This string of pearls then wraps up into larger coils. The coils wrap into even larger coils called supercoils. These supercoils are then looped and packaged with other proteins to form the condensed chromosome.
So this is how our chromosomes supercoiled. This causes the DNA molecules to coil back on itself, becoming shorter and wider. You can try it yourself, supercoiling an elastic band. So here the linking occurs with only a few twists, but to get width as well, which implies supercoiling, keep twisting here. So now we're going to watch about how mitosis actually occurs in the cell.
During interphase of the cell cycle, the genetic material of the cell is found in the form of chromatin and located within the nucleus of the cell, which is surrounded by the nuclear envelope. During the S stage of interphase, DNA replication takes place, and thus the cell leaving interphase and entering mitosis already has replicated its genetic information. In addition to the replicated genetic information, interphase cells that are about to divide also replicate their centrosomes. In animal cells, these centrosomes are formed from centriole pairs. As cells leave interphase and begin prophase, the first stage of mitosis, the chromosomes begin to condense. The nuclear envelope begins to vesiculate, and the centrosomes migrate to opposite ends of the cells to define the two poles. During prophase, the mitotic spindle apparatus begins to form by the polymerization of tubulin proteins. This creates microtubules that emanate from each centrosome. Chromosomes completely condense such that sister chromatids formed from the replicated genetic information and joined at their centromeres are clearly visible. During prometaphase, the nuclear envelope completely breaks down and the mitotic spindle is formed. Polar microtubules extend from both poles and overlap with each other. The sister chromatids become attached to the mitotic spindle via kinetochore microtubules that attach to the kinetochores, which are bound to the centromeres. As prometaphase is nearing its end, the sister chromatids are observed to jerk back and forth between the two poles. During metaphase, the sister chromatids become localized in a region called the metaphase plate. Once the chromosomes have aligned at the metaphase plate, metaphase is complete. When metaphase is completed, anaphase begins. The sister chromatids separate, and each chromatid is now linked to only one pole via a kinetochore microtubule. The kinetochore microtubules shorten, and the individual chromatids, now called chromosomes, are drawn to the poles. Toward the end of anaphase, the polar microtubules push against each other and cause the two poles to be moved farther away from each other. Each daughter cell will receive the same complement of chromosomes as was originally found in the mother cell, thus assuring that each daughter cell gets an identical copy of genetic information. During telophase, the chromosomes decondense and most of the microtubules depolymerize. Portions of nuclear membrane surround each individual chromosome, creating vesicles. The vesicles fuse with each other to create a nucleus containing all of the chromosomes at one pole of the cell. In animal cells, cytokinesis or cell division involves the formation of a cleavage furrow. The plasma membrane is observed to constrict and eventually separates the cell into two daughter cells. The daughter cells then enter interphase, where growth and enlargement occurs, leading to either preparation for another mitosis or differentiation and specialization occurs.
So now we start the mitosis processes. First phase is called prophase. What happens here? The centrosomes move to opposite poles of the cells and spindle fibers begin to form between them. DNA supercoils chromatin condenses and becomes sister chromatids, which are visible under a light microscope. The nuclear membrane is broken down and disappears. As you see here, centrosomes start moving toward the opposite poles of the cells, forming the microtubule organization. Chromosome consists of the two sister chromatids attached to the centromere, and nuclear membrane starts to break down, as you can see from the pictures. So the late prophase, what happens? The spindle microtubules, nuclear membrane has disintegrated. The metaphase is the second phase of the mitosis. The spindle fibers from each of the two centrosomes attach to the centromere of each pair of the chromatids, and contraction of the microtubule spindle fibers cause the sister chromatids to line up along the center of the cell. So this is how it's actually the metaphase looks like in the picture. So actually all the chromatids are actually in the middle of the cells. They are actually lined in the middle of the cell. So here you see identification of the mitosis in cells. Anaphase is the third phase of the mitosis. Here, the continued contraction of the microtubule spin fiber causes separation of the sister chromatids, and now the chromatids are chromosomes. A chromosome move to the opposite poles of the cells. So as you see here, how the chromosomes are actually separated to the opposite poles. And the last phase is the telophase, and how the chromosome arrive at the poles. Microtubule spindle fibers disappear already. New nuclear membranes reform around each set of chromosomes. The chromosomes uncoil the condensed to chromatin. So now cytokinesis begins. So remember these phases like that: prophase, metaphase, anaphase, telophase, like people, meat, and talk. Like people for the prophase, meat for the metaphase, and for the anaphase, talk for the telophase. PMAT, people, meat, and talk. Prophase, metaphase, anaphase, and telophase. So mitosis is the division of the nucleus, whereas a cytokinesis is the division of the cytoplasm and hence the cell. So let's look at this animation here.
Cytokinesis is the phase of the cell cycle during which the cytoplasm actually divides. It follows nuclear division and involves the splitting of the cell into two cells. In animal cells, cytokinesis is achieved by a constricting belt of actin filaments interacting with myosin. As the filaments slide past one another, the diameter of the belt decreases, pinching the cell and creating a cleavage furrow around the cell's circumference. Constriction proceeds, and the furrow deepens until it eventually slices all the way into the center of the cell. At this point, the cell divides in two. In plant cells, membrane components are assembled in the center of the cell to form the phragmoplast. These components are vesicles derived from the Golgi apparatus. The vesicles travel along microtubules, and their content forms the cell plate. The cell plate continues to grow outward until it reaches the cell membrane along the perimeter of the cell. Cellulose contained in the vesicles is then laid down between the new membranes, creating two new cell walls. Pectin is also deposited in the space between the daughter cells.
So, you know already that cytokinesis occurs after mitosis, and it's different in plant and animal cells. So, in animal cells, plasma membrane is pulled inwards with the aid of a ring of contractile proteins, actin and myosin. When cleavage furrow reaches the center, the two cells pinch apart, as you see here. But when it comes to the plant cells, vesicles are transferred to the area of the equator, and two layers of membrane are developed. Vesicles are transferred in the area between the two membranes, and the middle lamella is developed. The two daughter cells deposit cellulose on both sides of the lamella adjacent to the cell membrane, and the two new cell walls are actually formed. This is how is actually the cytokinesis occurs in the plant cells. So cytokinesis occurs after mitosis and is different in plant and animal cell, as you see here. So the mitotic index. Now we're gonna talk about the mitotic index. It's actually the determination of a mitotic index from a micrograph. What is a mitotic index? Is actually the number of cells in mitosis divided to the total number of cells observed, and by this way we can find the mitotic index. So count the number of cells in different stage of mitosis and then calculate the mitotic index. First, what we're going to do, we're going to count the number of cells in mitosis. Then we're going to count the total numbers of cells observed. Then going to find the mitotic index here. So now there's an exam question. Draw a label diagram of 4 stages of mitosis in an animal cell with 4 chromosomes. So this is how it actually happens. the prophase, metaphase, anaphase, and telophase. Don't forget to write down what happens in prophase. The chromatin condenses, chromosomes replicate, centrioles are at the poles, nuclear membrane degenerates. In metaphase, chromes line up at the equator, spindle microtubules attached to the centromere. The anaphase, chromatids pulled to the poles by spindle fibers. And in the telophase, the new nuclei are actually formed and cytokinesis begins. So today we're going to talk about the cyclins as well. The cyclins are involved in the control of the cells. The cyclins are a group of proteins that ensure that task in mitosis will happen on the correct time and cell advances to the next stage when appropriate. Their reaction is concentration dependent, and in high concentration they bind to specific enzymes called cyclin-dependent kinases, and this enzyme will in turn add the phosphate groups to other proteins, activating them. So let's look at the control of the cell cycle animation.
The process of cell growth and division in eukaryotes is called the cell cycle. This cycle is divided into phases based on what is happening in the cell at a given time. A cell grows during the G1 phase. During this phase, there is a chemical checkpoint that controls whether the cell will divide, delay division, or enter a resting stage. When conditions in the cell are right, the G1 checkpoint will be passed and the cell will enter the synthesis S phase. During the S phase, DNA replication occurs so that future cells will each have a complete set of the genetic instructions in the DNA. After DNA replication is complete, cells enter the G2 phase, where they continue to grow and prepare for cell division. At a checkpoint in this phase, the success of DNA replication is assessed. If all is well, the cell enters the mitosis, M phase. During the M phase, a complex series of events moves the DNA so that a complete set of genetic instructions will be sent to each daughter cell. The process of mitosis is assessed at a checkpoint during the M phase. Once this checkpoint is passed, the cell will complete mitosis as well as begin the cytokinesis C phase. Part or all of the C phase actually overlaps with the later parts of mitosis, so it is not a distinctly separate phase. During the C phase, the cytoplasm of the cell is divided, and two daughter cells are created from the original cell. When this process is finished, the daughter cells enter the G1 phase, and the cycle is complete.
So cyclins are involved in the control of the cell cycle, and there are 4 different kinds of cyclins. The cyclin D triggers cells to move from G0 to G1 and from G1 into S phase. Cyclin E prepares the cell for DNA replication in S phase. Cyclin A activates DNA replication inside the nucleus in S phase, and cyclin B promotes the assembly of mitotic spindle and other tasks in the cytoplasm to prepare for mitosis. Remember that cyclins are actually the proteins, and they are very important to carry out the interphase and the mitosis processes. So, cyclins, serendipity and scientific discoveries. Tim Hunt actually discovered the cyclins during a series of experiments on the synthesis of protein in sea urchin fertilized eggs, and he traced the protein that would be synthesized for 30 minutes and then being broken down. So he won actually a Nobel Prize for this discovery, and he has always emphasized on the effect of serendipity on his scientific discoveries. And he on his Nobel Prize lecture, he noted that he was lucky enough to select 10-minute interval in his experiment. So the selection revealed the true dynamics and the protein synthesis. If you want to read more, you can just click on this link and read more about the cyclins. So now we're going to talk about the tumor formation and cancers. So it's really prevalent among the human beings having tumors. But what are the tumors? They result from the uncontrolled division of a group of cells, and Tumor cells may stay adhered to the initial tissue and may not invade neighboring tissue, which we call benign, and most likely will not cause serious health problems. But in many cases, cells from the tumors travel through the blood and the lymph circulation, invade other organs and tissues, creating secondary tumors. These tumors are called malignant and may cause severe health problems. So these are different factors associated with tumor formation and the cancer. So the factors are known as carcinogens, and the air pollution, smoking, specific strain of viruses, different forms of radiation can cause to develop a tumor in the organism. In most cases, the carcinogens cause mutations. That's why they are also called mutagens. And mutations are the changes in the sequence of genes related to the regulation of cell divisions. So the correlation between smoking and incidence of cancer. A study was carried out in the number of cigarettes smoked by men per year and the number of deaths from lung cancer. The graph below shows the result of this study. Here, as you see, the cigarette consumption in the lung cancer. Here, you're going to describe the changes in the number of the deaths from the lung cancer between 1920 and 1975, explaining the meaning of the correlation, and describe the evidence shown in this graph suggests that there is a correlation between cigarette smoking and the number of deaths from the lung cancer. Here as well, as you see here, however, correlation does not always imply causality, but in this case of the smoking, 20 different chemicals have been identified to be causing tumors in lab animals, while another 40 have been identified as carcinogenic. So thanks for watching, guys. Have a nice day. Bye bye.